Hi all. In terms of preparing for the practical exams for your lab, think about the techniques that we did this semester, and I've listed out the techniques here for you to as a reminder. In terms of studying, I'd recommend that for each technique, think about why did we do that technique, and then go through the step-by-step process of the technique. Know how do you do that step, why do you do that step, and what would happen if you didn't do that step correctly. So for, let's say, using the scale to weigh things, that's a pretty simple technique. You use it to know how much something weighs. You do it by first tearing or zeroing your scale, placing your sample on top of it, and recording the weight of it. If you didn't do it correctly, maybe you didn't tear it, well, you'd be recording the wrong weight. All right, setting up hydroponics quadrants, right there, you took your media and pushed them into each quadrant. Maybe you needed a piece of cheesecloth in the bottom, so you filled up your quadrant with your media, you put your seed in and placed them inside, placed them floating on the nutrient media. The overall purpose of that technique was obviously to grow hydroponic plants. If you didn't do it correctly, say if you didn't have your media pushed down enough so it didn't touch the nutrient media, well, your media would not have any nutrient liquid flowing up into it, and your seeds would not be nourished and would not support plant growth. Things like plating microbes. So there you would have a liquid solution of microbes, and you had to vortex it. You vortex it to mix it up. You then pipette out a certain amount, place it onto your plate. You sterilize your spreader and then spread them evenly across the plate. If things that you might mess up, you can mess up most of those steps, and you might get contamination. If you didn't, say, sterilize your spreader, you might get an improper concentration of your microbes if you didn't vortex before you plated them, or if you pipetted incorrectly. The overall purpose is obviously to get a concentration of microbes that are capable of growing into a colony in your original starting solution. Sterile technique, what I mean by that is, what good practices do you use when working with microbes and whatnot? So think about there are possible contaminants in the air, on our hands, on the lab benches, on our supplies. So when we talk about sterile technique, to protect against things from the air, we work near a flame or inside a fume hood. To protect against our own contaminants, we wear gloves. And then to protect against contaminants on our materials, we try to minimize air exposure of all of our tubes and tips and whatnot, and we sterilize things that we can sterilize, such as the spreader. Serial dilutions, the purpose there is to go from something that is too high of a concentration to work with and do a step-by-step dilution process in order to get it down to a workable concentration. Otherwise, if you plated microbes directly from your gram of soil in 9 milliliters of liquid, if you plated 200 microliters of that, you'd have way too many colonies to count, and you can't accurately plate 1 microliter of that. So we do a serial dilution to get it to a nice concentration, so something that will have countable colonies at the end. Micropipetting, remember that we have 3 different micropipette sizes, so you need to be able to determine which size micropipette to use. You need to be able to set them appropriately. You need to get the tip on them. You need to depress the stopper down to the first stop, draw up the liquid, and then depress it down to the first or second stop to expel the liquid. Obviously, the purpose of that technique is to accurately measure very small volumes of liquid. Counting colonies is pretty straightforward. You count individual colonies on a plate. When you have a countable amount, ideally you want between, say, 30 and 300 colonies to accurately count them. If you were over that range, you could divide your plate down into little pie slices, count 1 representative slice, and multiply out to get the estimate of how many colonies were on the entire plate. UV exposure, remember when you exposed your yeast to UV, you took that plastic lid off because that lid potentially might protect against the UV light. So you then put your plate agar side down so that there was no plastic, no agar in between those yeast and that UV light source. Immediately after exposure, you put the lid back on and put them in a brown paper bag. We put them in that paper bag so that their photolyase repair mechanism would not be activated by exposure to regular light.